So, depending on who you ask, you may hear that the amyloid beta is the most significant factor in causing injury. You might hear that is the neurofibrillary tangles associated with the tau protein, or you may hear that they are simply artifacts of another, more important degenerative process that's occurring. So in other words, maybe they're just the remnants of the damage that's already occurred and they're not actually causing the injury. Unfortunately, nobody really knows the answer to this question. So here, what you can see is this is APP. So this is amyloid precursor protein. It's in the cell membrane, okay? And you can see that we basically have enzymes here, okay? So they're what we call secretases, so beta secretase, gamma secretase, etc., that actually will cut this A beta in different places to generate different beta amyloid proteins, shown here. Okay, so there's different fragments of beta amyloid that can be created depending on which secretase enzymes are cutting it, right? So there might be one that's 42 amino acids long, one that's 38 amino acids long, one that's 36 amino acids, etc., right? And there's actually a lot of research going on right now to examine all the different fragments of beta amyloid, so the different size pieces, to see if one is more what we call pathogenic than the other, or whether one is associated with more injury than the other. And what we can see here is that once we've got a bunch of beta amyloid around, because it's misfolded in some way, which can't really see the folding here of each individual one, but let's assume that it is, right, because that's what we think, is that it starts to stick together and aggregate into these big plaques, okay? And these plaques are found in the brains of Alzheimer's patients, all right? So here we can see this is a normal brain, looks very good. This is gray matter here, then we've got the white matter tract on the inside, and then we can see from this schematic here, this would be like the brain of an Alzheimer's patient. And so what we can see here is that the ventricles are larger, so you can see regular sized ventricles here, larger ventricles here, which really reflects some atrophy or some degeneration of some of the cellular structure around it, okay? And also the ventricles here. And we can also see that some of these brain regions are shrunken quite a bit. So here we would normally have, this is the hippocampus. So you know the hippocampus is a seat of memory, right? So it stands to reason that in a disease that's associated with a decline in memory, right, we would have injury to the hippocampal region. And here we can see a lot of shrinkage of this region here compared to the other one. And also some shrinkage of the cortical tissue as well. This particular schematic here, I think, is more telling. So this really shows us the difference between a healthy brain, age-matched control, and a severely damaged Alzheimer's brain. So this was from advanced stage Alzheimer's disease, right? Here, what we can actually see is the plaques that I showed you in the last picture. So this is your neuron, okay? And if we were to take a cross-section into this axon, what we would see here is these clumps or aggregates of this tau protein. So these are what we call the neurofibrillary tangles, all right? Now we have these microtubules, and if you recall from your cellular biology, or your bio class, we have microtubules that actually span the entire region here, and they help to move things back and forth and provide some structure in the axon. And here we can see that structure and movement is going to be very perturbed if we actually have these tangles of this tau protein here that kind of breaks up the microtubules and disrupts really the structural arrangement of it, all right? And if you see these two, what we call photomicrographs here, this shows staining of some common stains used in Alzheimer's studies, and they actually will show you what a big plaque looks like here. So you can see this big plaque and what some of these neurofibrillary tangles, okay? So they have special stains that will stick to the plaques and to the tangles, right? And they can see that. All right, so how do you treat Alzheimer's disease? There's a couple different ways that are being used to try to at least slow the progression of the disease, right? We can't cure it, not yet. One is an acetylcholine esterase inhibitor. So if you remember, one of the things that I mentioned is that a loss of cholinergic neurons is common. So if we lose the neurons that produce acetylcholine, we're going to have less acetylcholine transmission, right? And if that's important for memory and cognitive function, we're going to have some problems. So one of the things that we can do is actually block the enzyme that is responsible for degrading acetylcholine. And that enzyme is the acetylcholine esterase, right? So acetylcholine esterase inhibitors are drugs that can be given to bind up and block acetylcholine esterase, which will keep acetylcholine in the synapse for a longer period of time, right? Allowing it to act. Another one is memantine. So this is actually a drug that's main target is glutamatergic neurotransmission. All right. So what this really does is it actually blocks NMDA receptors. And so you'll recall when we went through the glutamate systems, we talked about a couple different receptors. We talked about an AMPA receptor and an NMDA receptor. They both respond to the neurotransmitter glutamate, right? And we had talked about amplification and NMDA being an important channel that gets opened in order to trigger a cell to fire. Now, the problem with overactivation of glutamate is that it can actually cause excitotoxicity. And so when we have lots of neurons dying, one of the things that happens is the neurons break open. And when they break open, they release all their neurotransmitter. And glutamate is one of the major neurotransmitters in the brain. There's lots of it around. Okay, so lots of cell death is going to lead to lots of glutamate being released into the extracellular space, and it's going to cause overactivation of cells that actually leads to cell death, right? Just because remember, too much of anything is bad, right? We want normal glutamate signaling in the brain, but we don't want excessive glutamate signaling. So one of the approaches has been to block some of these receptors that respond to glutamate to try to sort of dampen or decrease some of the excitotoxicity. Right? Excitotoxicity is just a word. It sounds complicated, but it's just a word that we use to describe when excitation of neurons is causing death of tissues. Okay, so excitation, right, would be like when glutamate binds the receptor and it causes the cell to fire. That's excitation. So excitotoxicity means toxicity that results from that happening, right? Okay, so again, we normally need glutamate for memory and for normal function, but we don't want too much of it around, all right? There's also some thoughts that memantine might actually stimulate acetylcholine receptors as well on cells through some secondary effects. So no drug is perfectly clean, and even though they're targeted to certain receptors, there's always a chance that they're going to bind to something else because that's just sort of what proteins do. And depending on how high of a dose you give, you might actually even get more non-specific binding the more that you put in there. All right. So that's essentially what's known for Alzheimer's disease. Now we're going to switch over to Parkinson's and talk about this for a few minutes. It's very different than Alzheimer's disease, even though they're both neurodegenerative diseases. Here on the right, you can see that we have the comparison between a normal dopaminergic neuron and a dopaminergic neuron in the brain of a Parkinsonian patient. And what we can see is the normal dopamine neuron is going to release lots of dopamine, and it's going to bind to the next cell, right? That's going to produce normal functions, one of which is movement, okay? So remember, we have these different dopaminergic loops. We have the mesolimbic pathway, and we have the nigrostriatal pathway, right? Nigrostriatal pathway tends to be more involved with movement, right? And so we can obviously modulate movements in this fashion. In a Parkinson's patient, we see there's very little glutamate, or very little dopamine. Yeah, we're onto dopamine now. No more glutamate. So there's very little dopamine being released here. And so what happens in Parkinson's disease is we actually have the selective loss of dopaminergic neurons. All right. Again, this is a motor disorder. Doesn't appear to be any kind of genetic link, so it doesn't appear to be heritable, but it is characterized by motor dysfunction. Right? And again, once we get to a certain point in the progression, we'll start to have some cognitive deficiencies and decline associated with it as well. But it's primarily known as a disorder of motion. Okay, so patients who suffer from Parkinson's disease typically exhibit tremors. Okay, so they're uncontrolled movements in the body, so they may have some shaking or some trembling. This may cause them to have difficulty doing normal types of functions that were usually pretty good at doing on autopilot. Things like being able to speak normally, being able to walk normally, pick up objects, right? And have really pretty good control of our hands and our limbs, right? This is something most of us take for granted, but in one who suffers from Parkinson's disease, this becomes very difficult to accomplish these sorts of tasks, right? Now, what's been found by examining, again, post-mortem brains of Parkinson's patients is that there is a specific selective loss of dopamine neurons, dopaminergic neurons, in the substantia nigra. So you remember that's one of the hotbeds, places where we have lots of dopaminergic neurons arising. Okay, so this is a hallmark of the disease. And it looks at this point, at least, to be linked to a deficiency in a particular protein. And this protein is called alpha-synuclein. All right. Now, alpha-synuclein is sort of a strange little protein because nobody was really very clear on exactly what its normal function is supposed to be. All right. So alpha-synuclein is found in the mitochondrial membrane. It's found in synaptic terminals of neurons. And it's thought that alpha-synuclein in these synaptic terminals helps to target vesicles, okay, for the release of neurotransmitter, and it's associated with that function. But we're not really exactly sure exactly how it works under normal conditions. But what we do know is that we have an accumulation, okay, of misfolded and misformed alpha-synuclein. So again, we're going back to the common theme of misfolded proteins, all right? So we have low levels of alpha-synuclein, or really, that is to say, low levels of the correctly formed alpha-synuclein. There's alpha-synuclein around, but the stuff that's around tends to be misfolded, and it's forming these aggregates again. So when we see an aggregate of alpha-synuclein, so again, this is like a plaque, sort of like what we saw with A beta, but when it's alpha-synuclein, it's found inside of a cell, we call it a Lewy body, okay? So a Lewy body is just another word for an aggregation of misfolded alpha-synuclein, right? And this is what's been discovered in these cells. Now, there's other forms of pathology of neurodegenerative diseases. Actually, one sort of general one is called Lewy body dementia. So it's not quite the same thing as Parkinson's disease, but there also is an accumulation of Lewy bodies. So Lewy bodies are not a thing that are specific only to Parkinson's disease, but they are one of the major features that's seen in the brains of Parkinson's patients, right? So if we look at this brain here, we can see this is a normal brain here. This SN, this means substantia nigra. So this is that region where all of these dopaminergic cell bodies will arise. And we could see here in the Parkinson's patient that there is a much different looking substantia nigra. Now these stains here basically tell us that we have a big region here where we have a lot of injury to this tissue. And if we look at zoomed in pictures here, we can see the normal tissue. This is a normal patient. And this is actually stained with TH. This is tyrosine hydroxylase. So if you remember from your biosynthetic enzymes, that's one of the enzymes that's needed to make dopamine. Okay, so we have lots of TH positive neurons, which means we have a lot of cell bodies of neurons that are making dopamine. Whereas here in the Parkinson's patient, look at that. It's virtually empty. Okay, so we've lost all of these different dopaminergic neurons in the substantia nigra. And here we can actually see a very zoomed in version, okay? And this is a Lewy body. So this is actually a collection of alpha-synuclein that's kind of plaqued together, all right? If we look at this image here, we can see a healthy brain. These red regions here basically show you where energy is being used. So healthy brains will show regions where there's energy utilization, right? They're using energy to do things, to perform cognitive functions as life goes on, right? And so they can measure this in the lab. And so we can see in the Parkinson's brain, we have a deficiency in energy utilization in these regions. Now, if you know about the brain, you'll know that these are the regions of, and it's hard to see here because this is a transverse or a horizontal section, and we didn't really look at a lot of horizontal sections. But this is actually that region that contains these dopaminergic cell bodies, okay? And so we have a loss of energy utilization in this region because these cell bodies have actually died away. All right, so the energy production is low. We also have some mitochondrial genes that have been identified as being